Okay, good afternoon, guys. Welcome to this image processing lecture. So first thing we need to understand the difference between the getting image processed using deep learning and why do we need the image processing uh, or image analysis methodology. So you have seen that when we apply different deep learning algorithms for some sort of classification, we say, here's an image and learn the association between the image and the class of an image, whether it's a tumor or whether it's a face of the face of a person or identify object in an image. So deep learning algorithms do learn those relationships, and they are good with the classification and learning that association. But the problem is they don't really tell you why they're doing this classification. And if you are a drug company, if you are in a pharmaceutical, or if you are in a research, especially in the medical domain, then you do want to learn, you do want to identify why these images are getting classified into so and so classes. Uh, so, for example, you've been employed in a medical setting where one pharmaceutical company or in a research institute, you are investigating the efficacy of a drug to the certain type of cell lines or cancer lines. You apply that drug, and then you feel you look at the images using microscopy methodology or advanced other type of methodologies. You look at the images and you see there's a difference before the drug application and after the drug application. You might see the cells have grown, you might see the cell has been shrunk. So it's not enough in a medical industry to say that, hey guys, here are the images where we have a small DNA, for example, and here are the images where we have a bigger DNA. Though we can see that with our eyes that yes, it's look bigger and this one looks smaller, but this is not enough. We need to quantify them. We need to quantify the images, and we need to say with the numbers that now this thing is bigger than, how much bigger than 10% bigger than 5% bigger, 20% bigger, or smaller. And then you also need to show whether this change in size or whatever you're measuring is significantly enough uh, to show the efficacy of a drug. So really the image, when we say the image processing or image analysis, we mean to quantify something. You would need to measure something in an image. We need to identify specifically a certain part of an image called region of interest. So that's why, especially in the medical industry, the image processing is a very popular like methodology, and every data scientist should learn it, apart from implementing the state-of-the-art deep learning algorithms. So when we say the image analysis, it means the extraction of a meaningful information from images, mainly from digital images, by means of digital image processing techniques. So these images, these techniques, some of these techniques are quite old compared to the deep learning methodology that we have nowadays. But still they are getting used, implemented, and heavily used in an industry. So let's look at few case scenario coming from my own experience. These are some of the images that I've taken while I was working for the Children Medical Research Institute in Australia. And we were looking at the uh, the role of proteins and their co-localization with the telomeres. Now, what is telomeres? I know you will not know it. Telomeres are the end part of the DNA. So if this is, look at the left side of the screen, this is our chromosome. So these end part, the green, these parts are telomeres, end part of the DNA. They are not genes. And why they are very important? Because they are involved in about 80% of different type of cancers. and they are also have very important role in a lot of childhood diseases, such as aging and others. So a lot of research happens, and especially the institute I was working at the time, uh, one of their specialty was to do research on telomeres. So here, uh, different proteins were interacting with the DNA, and then those proteins were stained on this one, on these, the first red channel, we have a protein that's been stained red. So the dots that you see here, these are the protein. And similarly, on the green, there's another protein which is stained, and then the nucleus itself is being stained as blue, captured on a separate channel. So now these three different channels are captured on a microscope, and then we look at how these green dots and green dots and the red dots are interacting within the nucleus, right? And if you look at the RGB, the far right part of this slide, so you can see that the red, when we overlay them, red and green, within the blue, within the nucleus, so you can see lots of interaction happening. They are right on the top of each other, but not everything. So like, look at closely, this green doesn't have any red on the top. So now these are three different cell lines. This is a different cell line. This is another cell line. This is another cell line. And when you look at the RGB of these images, you'll see that interaction happening. So that means, just looking at these images, you can see that yes, there are some interactions happening. They are right sitting on top of each other. That means they might be interacting with each other. They might be bound to each other. They have some roles with regulating these telomere phenomena. But that's not enough, yeah, because you can see that phenomenon happening in three of the cell lines. So what's the difference? So we need to quantify that to say that maybe this is happening more here, maybe this is happening less. And even just saying more or less is not enough. We need to quantify exactly. So this is discuss the whole process in this research paper in the scientific reports. People who are interested, they can read more. In the second use case scenario, again, working in the same Children Medical Research Institute, we were looking at the length of the telomeres, because the length of these telomeres, the DNA end part, they are also they also tell you how long you're going to live, right? When you're going to age. So the longer the telomeres, that means hopefully the longer you're going to live and the less chances of getting the diseases. And in many childhood diseases, what they have observed, that the children that have a shorter telomeres length, they develop certain type of diseases, right? So one of the mechanism is to measure their telomeres and then see the likelihood of getting that person disease. So if I put myself on the left side of the screen, so so here I designed this automatic algorithm which automatically identifies. So all these are noise. This is all noise, not meaningful. The meaningful information is this one. So one of the process that you're going to do during the image processing is the getting rid of this noise, because this noise is experimental artifacts. You're not interested in that. The interesting thing is only this one. These are the things. These are the actual telomeres that you want to measure. But the problem here, you can see there are some gaps there. So visually, when we look at them as a human, we know this is a single piece of DNA. But because it looks broken because of the experimental artifacts, we need to join them to do the correct measurement. And I designed this sophisticated algorithm that basically joins them and that do the correct measurement. Again, that's not using any deep learning or machine learning. That's all image processing method. And in this third research work, we again looked at the efficacy of a drug on the cancer cell line. So this one are untreated. So this is the spindle of the cell, And here's we have blue as a, this is the DNA, and then the right part is RGB3 images. So the information is captured on 3 channel. One channel is a spindle. So people who come have a little bit of biological background, they know when the cell divides, this spindle formation happens. The DNA get duplicated, and this spindle pulls this DNA apart into two, and then 2 cells are formed. So cancer, as you know, is nothing but just a uncontrolled growth of cells. So your cells start to grow more and more and more, and you get a lump, and then it becomes so big, and then it start to create a lots of problems. So if we can if we can see how can we kill these cells, or how can we stop that uncontrolled growth, we can solve the cancer, right? So in this experiment, one process was implementing, where we were knocking out this gene. So these are the untreated one, and then the bottom one are the treated one. So now, if you look at as a human, you capturing that these images on the microscope, you can say, or this is this spindle looks from different from this one. So these two not supposed to have any difference, right? So without going into too much detail of this especially this work, but just to give you an idea that these two supposed to be the same. Again, these two supposed to be the same. So there's supposed to be a difference between these two groups and this group. But if you can just look at these spindles and these DNA, even these two are different. They're not exactly the same, right? And this one looks a little fatter. So now you can say, okay, the DNA is a fat, is more thick, it become bigger. But then how big is that? Even this one looks a little bit different from this one, but there's supposed to be no difference between these two, right? So that's where the image processing comes in, and you do the measurements of these two DNA, and then you show that yes, there's a difference, but that this difference is not significant enough, whereas this difference is really a significant enough. So that means that this drug has an effect and is doing something to these cells. Again, described in this research paper 2017. So now let's get started, and we're gonna do some work on this data today. So for that, we need to cover some basic concept of image processing. So the first concept is to know that every image can consist of one channel or usually 3 channels called RGB. R stands for red, green, and blue. So, so these 3 channels, when we say channel, basically, so that means it's just a 2D matrix. So red channel is a 2D matrix data. Similarly, green is a 2D matrix data, and blue is a 2D matrix data. So when we create an RGB image, basically what's happening, these 2D matrices are put on top of each other. And each pixel has a value, it's just a number. It's just a number there. The bigger the number, the brighter the pixel is, right? So the maximum value that a pixel could have is a 255 generally. It could be much more than that, especially for the scientific images. But generally, images can have intensity between 0 to 255. And the bigger the number, that means the brighter the pixel. So that means if it's a 255 here, that means the red is a maximum, and having 0 mean is black, no red there. And every RGB image is made up of these 3 channels, right? Any question so far? So in research, you might be given only a 1 channel. You might only receive just a grayscale image, and that's fine, right? So you don't always get a RGB images, but generally in a normally whenever we're doing the image processing, most of the time we get an RGB image. So a single channel image is called a grayscale image, right? So where each pixel is a shade of gray. So this is all black, mean the values here would be very close to 0. Either it would be every pixel here has a value either 0 or very close to 0, like maybe 1 or 2 or 3. And the pixels that are here showing a spindle, and they're supposed to be the DNA in the middle. So this spindle must have a value higher than 0. It could be 5, 10, 20, 30, 100, because they look brighter, right? Another type of image that we usually create ourselves, we don't get that. So we are data scientists are usually either being given a grayscale image, or they would be given an RGB image. So one of the image type that you will be creating is a binary image. So where you will create exactly the same size image like before this one, but each of the pixel here will either have one of the two value, either it's a 0 or 1, black or white. Again, it's a single channel image, and we use this to mask for the, we'll look at that in more detail. So binary images are very important because they allow us to do some sort of measurement, look at how much the perimeter is, how much the mass is, and also they help us to extract the region of interest, because we kind of define the boundaries in our binary image, and then we go back to the real image and extract information from there using this mask, right? So we usually come across various type of file types: JPEG, GIF, PNG, TIFF, BMP. You have seen them. But in research, the best images are the TIFF images. Whereas the JPEG and the GIF and PNG, they are compressed format and they lose the quality. In research, we also have many other types of file formats, and many are proprietary file formats, but usually all the proprietary formats convert files into finally TIFF format, and TIFF is the best to process, to do final image processing, but only for the final image processing. But usually what happens with the TIFF, because the file is not compressed and it has an original quality, it has original pixel values, but the size of the files are very big. So if you apply any deep learning or image processing methodology, it's going to take long time. You might have thousands and thousands of files, very big files. So if you process them, it's going to take ages. So usually what I do, and it always worked well for me. It's not a standard protocol. I'm not saying that this is a general rule of thumb. This was just my practice, and it worked well for me all the time. So what I used to do, convert an image into the smaller size JPEG image, develop all the algorithm on these JPEG images. And once my algorithm, once my format and everything, my pipeline is perfect, then I apply it onto the bigger original TIFF images. Because processing on original TIFF images take ages. So you'll be wasting lots of your time, your computing power. So, and for me, it always worked well. Whatever I designed on the smaller size JPEG images, that worked well on the higher TIFF images as well. So this is the piece of code we're going to do that practice today, where you read a folder, you read all files of the folder, and you convert them into the JPEG format, right? JPEG or whatever, you can convert into other formats as well, but usually JPEG is a good, it's a very small file size, easy to process, and easy to get your pipeline like working. Okay, somebody has their mic open. If I can request everybody to check their mic and do that. Now, what do we do with the image processing? We usually do 3 types of common processing when it comes to the image processing. We be either enhancing the quality of images. So your images might have a blur in there, they might have a noise in there, they might not be having a good contrast in there. There could be an issue with the they might not be very bright, the information might not be very well defined. So we improve the quality of overall image uh, for two reasons. That could be just for visual reason. For example, you might be employed in a company where they developed an app, and whenever you take a picture using their app, you want to have it processed, and you want to look it nice. In different apps nowadays, Facebook and other, they have this different type of filters that you apply onto the image to beautify your image. So um, that kind of work could be called the image enhancement. And you also do that in a research as well. So you sometimes improve the quality of an image before you feed into the real pipeline. Okay, so maybe I need to look up who's got their mic open and then mute them. Okay. And then we could have an image restoration task where you might have an old images from olden days, and you want to restore that. Lately, you might have seen some movies being reprocessed and restored from the olden days, even the original moon landing images and the pictures and the video has been much more improved now, and it looks beautiful. And even many olden days movies, the black and white movies, they also been colorized using this image restoration techniques. And you could also have some sort of issues with an image, and because you can't really go back and take those images again, because it's going to cost you a lot of money, time and effort, so you try to restore it using the image processing methodology. And then in the image segmentation, the most important thing in image processing and in a research data set, that you segment the region of interest. I'm going to use this word again and again, ROI. Please remember that, memorize it, ROI, region of interest. So in a research, in most of the application, you will have a certain part of your image is important than the other. For example, you're doing the facial recognition. You want to identify somebody out in the wild. So what is the most important part? Are their hands, legs, or what? So the face is the most important. Whenever you want to identify, so you're somebody, so your region of interest would be the face, eyes, nose, mouth, this area. So if we are studying the movement of cell or the efficacy of a drug, or its ability to kill the cell, so your area of interest could be DNA, could be spindle, or a certain organelle in a cell, right? So most of the time, the region of interest is very well defined. Now you need to identify that where exactly is that in your image, and you want to extract and then quantify its properties. So usually we do these three things, and today we're gonna look a little bit of all of these one, and especially we'll do a practice on image segmentation. So when we do the data analysis tasks, this is my own kind of pipeline. It's not a general rule of thumb. It's just for the practice that we're going to do with these images, where we're going to look at the size of a spindle or the size of these DNA. So usually in the research, all type of images have some sort of noise. So usually the task starts with removal of the noise, and you get in these images, this is all noise. This is everything that is shown here. This is all noise. So our region of interest could be this DNA or this spindle, right? So then you extract this region of interest, and then you do some quantification. You might need to do some image enhancement. If we're doing the image enhancement, now this is a very important point to remember. Like, look at this image from here to here. This image is being improved, enhanced, and this depends now why we want to enhance this image and what sort of quantification we're going to do. If we are doing any sort of intensity based analysis, then this image enhancement is no good, because we really want to capture the intensity of a region of interest, because this intensity is a representative of something that could be due to the low level of protein in that part of the image, right? So usually the presence of proteins when they are stained in a cell, the more the protein, usually the better the staining. So that means the intensity will be high. So intensity-based analysis uh, require you to process the images in its original format. Like, then you don't improve the quality of the images, because when you improve the quality of the image, basically what you do, you change its intensities, you enhance or decrease the intensity, or you modify the intensity. That's what you do when you enhance an image. So remember, whenever we doing some intensity based analysis, looking at the quality of the pixel, then we don't enhance the image. And then for all type of measurements, we need to binarize the image. We create a binary image, a mask, and then use that mask to extract the ROI from an image, and then use that a binary and ROI to do other sort of calculations around that. So we're going to do that all today. So now we'll look at some of the common morphological or common steps that you do in the image processing, right? And one of the very famous and common processes that you do on during the image processing is morphological operations. And there are two morphological operations. One is called dilation, another one is called erosion. Yeah. So both of these processes require a structuring element. So what's the structuring element? Remember when we learned this convolutional neural network? So we said we use a small kernel or filter. So it's the same thing here. So here we call it structuring element. So a structuring element is a small, just a 2D matrix. If you're processing 2D image, it could be 3D element. So basically for a 2D matrix, for processing one channel, you have a small matrix. It could be 5 by 5, it could be 3 by 5, and it could have a different shape. Like this square 5 by 5 element, there everything is one. So in a diamond shaped 5 by 5 element, you see some pixels are 0, and then basically creating a diamond shape. Like this one is a cross shaped. Now you see this one is a 1, all the rest are 0, and this one just a square 3 by 3 element. So using these different structuring element, then we use it to process to the original. So the overall concept is exactly the same as the convolutional neural network, like how we do that, the convolution over there, but the calculation is different, right? How do we calculate? Here's the calculation. So here we only have two type of morphological operation. One is called dilation. So dilation mean you making things bigger. Like when you dilate the syrup, what do? You put more water in it, right? So dilation mean you making things bigger. So this is your structuring element. And this is a very simple structuring element where we only have a one row and 3 pixels in there, and each one of them is 1. So what you do, you use that small structuring element and then move it across to all the input image. And if it's a dilation operation, what it's gonna do, it's gonna look at the neighboring pixel. And if the neighboring pixel is 1, it's gonna make it 1 as well. So that's why it's called dilation. It's increasing. So if this is 1 and your structuring element neighboring 1, even if it's a 0, you're gonna get 1 here. So that means now your thing become bigger. And if it's that was a binary image. If it's a grayscale image where you have a numbers in there, the structuring element will always be in the form of 0 or 1, right? So now you have a value here 16. The next neighbor pixel was 14, but because the structuring element here has a 1, so it's going to make this 14 as 16 as well, right? So you increasing the intensity in a grayscale or in a binary image, you making the size bigger. So that's why it's called dilation. On the other hand, erosion is an opposite operation where you're making things smaller, eroding. Yeah, so erosion. So it's the same. It looks the neighboring window and neighboring pixel. And if the neighboring pixel um, um, is 0 in your structuring element, it make your that pixel as well the 0. So here's an example. If we have these two objects in our binary image, so look, they are not joining, so they are separate. But if you apply a dilation process, so what the dilation does, it increases the size. It it increases the number of ones around your object. So this one becomes a little bit bigger, this one becomes a little bit bigger, and the both gonna end up making a one object. They become joined. So you also do this dilation operations to join two objects which are very near to each other. And this is what I did. One of the things I did in this work, where we have a breaks. Yeah, here's a break. Yeah, so this break was a partly being joined together using the dilation process. And I use another method as well, which I don't go into the detail, but one of the things I did is this one, dilated the object to join them together, right? And if it's an erosion process, so here you see you have two objects. Now you do the erosion. So erosion, what it does, it looks all the surrounding image, and because the surrounding pixels are zeros, so it makes your neighboring pixel 0 as well. So that means the sizes become smaller. So this object become very, very small, and the object that was already very small is completely gone. So now you use the erosion operations to get rid of the noise. So if this was a noise, so now it's gone from your final image. And then you can again apply onto this image a dilation. And this object almost will look similar to the size before. But now doing erosion followed by dilation, now your noise is gone. So this is one of the methods to get rid of the noise in your images. And another thing, you also do the erosion to make a fine boundaries around your object. Yeah, making sense? If any question, you can open up your mic and ask. Okay, now I've been keep using the word neighboring element, neighboring pixel. So how do we define a neighbor? Yeah, it's based on how we set our parameters. So if we say we only gonna look at 4 neighborhood, so that means we're gonna look at vertically and horizontally to our neighborhood. So these pixels are not defined as the neighborhood because they are diagonal. If you say that I'm gonna look D neighborhood, that means you only look at the diagonal pixels to define the neighborhood. Now these orange pixels are your neighboring to this pixel P, and these are not your neighbor. Yeah, because we define the neighborhood based on diagonal. And if you say 8 neighborhood, that means that everything around this pixel is a neighborhood, right? This now all is a neighborhood. So that means we have to define it. So why do we need to define? But because when we're doing the dilation and erosion, in both of these cases, we need to define the neighborhood, whether we're gonna just dilate to horizontal, vertical, or just in a diagonal or in all directions. So both of these operations require you to define your neighborhood. And these are the three ways by which you can define your neighborhood, right? Usually we go by 8 neighborhood, but that depends on the application. So again, this is also called object connectivity. Object connectivity also get defined based on the same concept. If the two objects are set to be connected based on whether we define their connectivity for neighborhood, for connected, or 8 connected, or D connected. So in this example, if we say we only defining connectivity based on the 4 connected, so that means for this pixel P, these grains are the connected object. Any object that is here is connected to the P, but any object that is maybe here is not connected to the P. But if we say 8 connected, then both of these objects will be defined as connected object. So why that is important? This is very, very important in all of the applications that we discussed here. In this example, dilation or erosion. Even in this examples, when we were extracting the DNA, and also in these examples, and also in the other examples here, because we are measuring the length of the telomeres automatically, and it's all based on connectivity. What's being considered as a noise and what's being considered as a real telomere. And also in this one, where we're saying this red is right on the top of the green, so that connectivity again based on that, looking at how you're going to define that. Does it have to be right on top of each other, or could be 1 pixel apart depending on diagonal or horizontal or vertical? So that's why these are very important parameters to understand and to implement and to define how you want to define, and also explain it to your client, whoever has given you the image to be processed. Now, we usually during the image enhancement, we have to all the time look at the image denoising. Most of the time, images have some sort of noise in there. And one of the most common type of noise is a Gaussian blurring, Gaussian noise, where the noise have some sort of bell-shaped curve. Or other type of noise is salt and pepper noise. This is, usually in old pictures or old images, you usually see that salt and pepper type noise in there. So we can get rid of these noise, improve the quality of the images by different filters, and median filter is one of the famous filter. Other type of more sophisticated filters are adaptive filter, veneer filter. So we're not going to go into the details of these advanced filters. They are available to you as different methods when we get into the code. But just to get an idea how these filter works, if we look at the median filter, so what it does, we define a window, and we say it's a moving window, and we define that we're going to take the median number from this window, and then you move this window across the image. And if you have an average filter, you replace the value of every pixel based on its neighborhood values. Could take an average. So these two filters are easy to understand, whereas the veneer and spatial filter, they are much more sophisticated. They have a complex mathematical formula. But basically the concept is the same. If this is your image, and you have some values here, so then every pixel, for example, 3, 4, 5, 7, 9, 3, 2, whatever, something. Yeah. So basically, then you define a window. You say, this is my window 3 by 3. You move that window across. If it's a median window, you look at the median. This is just like altogether, these are 9 numbers. So look at these 9 numbers and see what's the median in there, or take an average. In the average filter, you're going to take an average, and then the value of every pixel get replaced by its average, and then you just move this window across, and this solves many. Like in these images, I've tried myself. Salt and paper, the median filter works beautifully well, and it automatically gets rid of all this noise. Now, another concept is about dynamic ranges of your pixel values. So when you take a picture, and you would have observed that whenever you're using your own smartphone camera to take the images, sometimes some part of your image become really, really bright, and the other part are really, really dark. So in that case, the image doesn't look really good, and maybe you can do so much with these images. So in all of these, we have these four images here. So we're going to say that in these first three images, the dynamic range is really high, because you see there's a very high pixel values are here, maybe close to 255, and then you have lots of pixels with very like low numbers. And similarly here, few pixels with a very high values, and most of the pixels are with very low numbers. You can hardly see anything. So what you really want to see is a range compression, where you have a um, some range of pixels. Like you see how beautiful this image is. You can really see each and every part, and you can do a lot with this type of images, because your information is not saturated. And then whenever you're doing the research image processing, you have to define your boundaries very precise, because it's all about getting things calculated, quantified accurately, right? But there's a problem with every type of microscope or any type of camera system. So whenever the light goes on to the lens of your whatever the camera is, it has a point spread function. Like your object, like we know that every object has a very well defined boundaries, right? But when you take a picture of it, you zoom it, you can see the boundaries will not be really clear, and they your boundaries will always have this kind of uh, fuzzy boundary. You can say the fuzzy, right? So it's because of the diffraction of the light, because that's how the light works. So that means every system, every microscope in the world, every camera has this problem of a point spread function. We just need to be aware of it. And Microsoft manufacturer gives some values, some numbers, and that's how their price also get determined, that how good they can capture the object and define this point subtract function. The minimum, the better. Yeah, but it's always there. It's just to be mindful of that, that if the actual object is like that, obviously every object has a very clear defined boundaries, but when you're gonna take a picture or image, it's gonna look something like this. Right. And that, so somehow we need to learn what the point spread function is and get rid of it to accurately define the boundaries of our object. Right. So that's why it's called convolution. So convolution mean it's been mixed up, as you know, like convolutional neural network, you mix a filter within your image. So the same thing here. The actual object is get mixed with this artifact, with this point spread function. So what you need to do, you need to deconvolve your images. Many times your images will have some sort of noise. So this is considered one type of noise. Another type of noise is here, salt and pepper. It could be Gaussian noise. there could be many type of noise, right? So many times you will be required to denoise your data, denoise your images, and this is called deconvolution. So here, what we're showing here in this one, the middle one, the left one is your original data, and in the middle one is your noisy data. You can see this is a clear image, and this is a little bit of noisy image, and we somehow want to restore an image. We want to take this image and bring it back this image, right? You apply different kind of method. Here's a one famous method called Richardson-Lucy method. But now even deep learning methodology are very like sophisticated. We have sophisticated algorithms. So you can take these images, and you can train your deep learning algorithm to bring back this image. So it automatically learn the noise and bring back the images very close to the original data. Okay, so now we're gonna look at one of the most important tasks of all the research type image processing, and it requires you to extract the region of interest. Yeah, this is called region of interest segmentation. There are many methods out there, and one of the most famous, most common method is by thresholding and using a histogram, right? So remember what the histogram is. If we say frequency plot, density plot, histogram, they are all pretty much the same thing. What do we mean by thresholding? Thresholding means we create a binary image based on the grayscale images and define a certain boundary. And after that boundary, anything become over that boundary, anything become 1, and below that boundary, anything become 0, or the other way around. So if your pixel value on a certain location is greater than t, so for example, like we said that our images have a pixel value between 0 and 255. Each pixel can have a value between 0 to 55. 255 mean like white, 0 mean black, right? So maximum value, depending on which channel is that, if this is a red channel, the intensity of that channel, or blue channel, or gray or the green channel. So now if we say that our threshold t is 100, so then we say, okay, any value greater than 100, then you change that pixel to 1, and everything lower than 100, you change that to 0. So it's going to make your binary image, right? So as we discussed, the binary image can only have two values, either 0 or 1, or it is a black and white image. You don't have a grayscale values in there. And these are really important image types, because you convert the binary image and then you use these binary images to do a lot of other stuff, right? So that's a very simple thresholding. So you come up with a number, and you say that anything above that is 1, anything below that is 0. Now, how do you come up with this number? You're gonna come up with this magic number by looking at the histogram. So what's the histogram is? You take your image and you look at the value of each of the image, and you bin it, and then you do this plot, this count, like if this is 1, this is 2, this is 3, this is 4, so on and so forth. So, and at the end here you have a 255. So on x-axis you have an intensity of your pixel, right? On y-axis you have a count. So here, let's say if this one is 100, and this one is, let's say, 5, this one, let's say 10, this one is 15, this one is 20, let's assume. So we're saying that there are 5 pixels having a value of 100, right? Let's say this one is 50, So, and let's say this is maybe, let's say 200. So basically, this histogram is showing there are 200 pixels of value 50, right? So if we expect to see there a number of regions, that usually there's this kind of gap. So that's where our threshold is. So in these images, our region of interest is this gray area. We want to extract this gray area. If we do the plot of this histogram, so we can see that that's where it breaks. It could be 110. And then we can say anything above 110 is 1, anything below 110 is 0. So it's going to make a binary image exactly extracting these region of interest. Yeah. And remember, looking at the histogram, we cannot reproduce the original image. All of these three images, they are, as you can see, looks entirely different, but they have a same histogram, because 50% of the pixels are gray, 50% are white. So their histogram looks exactly the same, but you can see the images are entirely different. So you cannot reproduce image from looking at the histogram, but from every image you can calculate histogram, and you can automatically extract this minimum point of the histogram. So you take any, this is called the global thresholding, because you come up with this point where you have lots of points of background, and you define that this is your object of interest or region of interest. So then you can define these boundaries based on this certain grayscale values, and then you can set your thresholding. Now, the issue is, if you have this type of image, this one, where you have your background is not really consistent. So here our region of interest are these rectangles. We want to extract these rectangles, but the backgrounds are very different. If you apply a global threshold anywhere, either you apply global threshold here, or in the middle, and if you extract, you're going to miss out some of the rectangles, because it's all because of the background. It doesn't matter where you apply that threshold, you will miss out the some background. So in this type of situations, we might need to have many thresholds depending on the area. So this is called local thresholding or adaptive thresholding, where you define certain areas and then you apply a different threshold on that. So this is what I did with this work, because here our region of interest was this DNA, and there could be another spindle here, there could be another spindle here. So I defined an area of interest, and then worked out the thresholding just within this area, and ignore everything else. So this is, if you have a complicated background, this is what you can do. Just somehow define your regions based on, you might consider, and that could be experimental artifact, that you have this problem in a microscope, or because of the certain experiment, that always there is a bright background here and there's a dark background here. Then you can define an area where you could have a threshold maybe a little bit higher, and this is an area where your threshold could be a little bit less. So that's why in this one, these images, in first area with the threshold being used 80, in the second one 128, third one 188, and the fourth one 226. And you can see with this one we were able to extract all of the rectangles. Though there are still some noise there, but we can get rid of them easily by erosion or some other methods. Yeah. Any questions so far? Now, why do we need to do the binary image? Because actually the binary image allows us to define the boundaries. If here our region of interest is identifying people in an image, so if we identify that using a black and white image, that means we don't need to look up everywhere else. We now targeted this area, then we can apply whatever algorithm we want to apply. That could be deep learning, conventional techniques. So now this is our region of interest. We defined that. So all type of surveillance system, missile system, all sophisticated things that you hear about, automatic cars, all these equipment, that's what they do. They define their region of interest by these binary images, and then they target their application onto that area. Okay, now in research is also very important to have your image being appropriately exposed to the right light conditions. And you are the one who gonna let your scientist, bench biologist know that your images have a problem. Look at all these images. All these three images looks fairly good to work with, not really a big problem. But this one, the C1, is overexposed. How do we know that? We're going to look at the histogram. So you create an histogram from the image, and then you see there are lots of pixels that have a maximum value, 255. So you never let your pixel touch that maximum value. These are called, in research, these are called saturated value. So that means you don't really know what the actual value could be the pixels could have here. So all the microscope, all the sophisticated machines, they have the knobs on their machines in their software where you can do some adjustment and you can avoid this situation. So if your image, if there are like only 1 or 2 pixels having 255 value, maybe that's not a problem. But if it's more than that, in a perfect scenario, there should not be even a single pixel having an exact 255 value. Everything should be below than that. If that happens for a quite number of pixels, then your experiments are overexposed. They are getting a lot of light. Now, in an underexposed situation, you see a lot of pixels having a lot of um, like the count, the values are really high here, and then the certain part of the image really going up. So what you really want to look at in a good image is a really like a nice distribution of pixel values around that range. Right, but sometimes is something that you can't really like. I would say this, I'll show you when we jump onto the Jupyter notebook, that these are the underexposed, these images are underexposed, but we were still able to work with them. uh, it happens. Sometimes you can't really solve an issue experimentally, but then you get what you get and then you work with it. So we were able to salvage the images from these underexposed situation, and we'll look at in our Jupyter notebook shortly. Right. So in today's Jupyter notebook, the practice we're going to do is using this research paper published in 2017, where we were looking at the efficacy of this experimental procedure where we knocked out the clathrin heavy chain protein. And we know that this protein being heavily involved in the cancer. If you take that protein or take that gene away from your cancer cell, so then cancer cell will likely to die. So to show that, you do this experiment, and we calculated the DNA and the spindles, and we show the efficacy of this experimental process. So I've got these images uploaded, and actually the people who might be watching on the YouTube, they can get that images on this on this from this article as well. Okay, so how do you know that you have done a good image processing? So people are going to give you the images that here's my images for one treatment, here's my images for the second treatment, here are my images for the third treatment. And they expect you to show the difference. All the studies tend to produce positive results. All studies want to show that their drug, their process does have an effect and it does solve the problem. And if you don't give them the answer they're looking for, then probably they might say, oh, you don't know much of your work, you're not a good data scientist. So they might push you to continue to apply different algorithms until you give them the answer they were looking for. So as a good data scientist, you don't really do that. You show them what you should, what really is present in your images. So we as a data scientist, we don't hide the dirt under the carpet, and at the same time, what's visible, we just try to show that pattern was in the data. We don't create the pattern. We just highlight the pattern that's available in the data. Right? So I hope this makes sense. So how about we take a break and then we jump onto the Jupyter notebook. Okay, now let's look at this Jupyter notebook. Here we have, so first of all, let's get rid of this one. Okay, the first thing is we load important libraries, and the most important libraries we're gonna use today in this exercise are matplotlib, obviously, skimage, numpy, and few others which we're gonna load later. So let's run this one. So the first, I'm gonna explain that the image is nothing but a 2D matrix. So the first line of code is generating numbers, generating a 2D matrix 10 by 10, and generating some numbers randomly, right? So, and we're printing here that is a float value, is a 10 by 10 matrix value, and we just show it. So any matrix, 2 by 2 matrix, any 2 by 2 matrix can be shown as an image. So you put the matrix here and you put the C map equals gray. That means the color map is just a gray, is a single channel, and it's going to stretch the values to show the color difference between the pixels value. Though the pixel values here are between 0 and 1, because that's how the random number get generated. But this color map tends to make it spread out the color values basically. And I'm showing the value for the first pixel, right? So print rand 00 mean we're showing the value of the first pixel. The value of first pixel is 0.8. So bigger the number, the one is a scaled value, right? Remember the 255 now represented by 1 here. So I run it again. Now the pixel value is 0.08, very close to 0. So that's why it's very dark here, almost black. And then I want to show you how you change a pixel value. Now remember, now our image 2D matrix is called RAND, R-A-N-D, RAND. So what I'm going to do is make the first pixel, which is a black here, I'm going to make it 1, right? And then we look what it looked like. So you see now this pixel is now shown as white. Why? Because I changed the value, which was previously 0.08 to 1, and now it's been shown as white, right? So two learnings here, that the pixel, each color pixel value is just a number, It could be a number from 0 to 1. And if you remember during the convolutional neural network, when we were doing the training, in that exercise, we brought the numbers from 0 to 255 to the range 0 to 1, because that's good for the deep learning. Here, because the random number are getting generated from 0 to 1, so we don't have to do that. So a number could be anything. And in a research environment, these numbers could be like 16,000, 20,000, 50,000, doesn't matter. You can always scale any number between any range. So the main concept here is image is made up of just a numbers, right? There's a certain range that you always know what the range is. That could be from 0 to 1, and all type of images could be translated back to this basic range of 0 and 1. And this is what we do during the image processing and all deep learning applications. We always convert them from into the range of 0 and 1, right? And you can, another thing you learn here, that the pixel values can be manipulated individually. So you can take one pixel out, change its value, put it back into the image, and that's how you can manipulate any pixel in your image, right? Then the exercise 2 is changing the image size and convert images to another format. Remember, I said I like processing images into the JPEG because the original images that I was given was in TIFF, and they were very big images, bigger file size, and it takes ages to process those images. If you are in a similar situation, then it's my like personal advice. That's not a standard rule of thumb, that you change those images into a smaller size JPEG images, get your pipeline, get your code working right, and then you apply that code and algorithm onto the bigger images. You could have a like 1,000, even for your assignment, you might have a 10,000 image to process. So if you, every time if you apply things to these 10,000 images, that could take hours and hours, right? So you can take just a 500 images, convert them into the smaller images, small size images, and then have your code pipeline and everything working. Have a look what the images look like. If you're happy with all the code and the pipeline all working, then you apply it to full size images. So this is just showing the execution of the code from one format to another format. So here's the extraction of region of interest in this biological system that we are working with. I've got two folders. One is called untreated, another one is called the CAC. We can have a look here. Yeah, it's a one untreated, another one is a CAC knocked out folder. So we load one image into this variable called img, right, run. And we show it, plot plt matplotlib.imshow, and you do that, specify your variable here. So, and we showing the CMap is a grayscale. So the original image, if I take this one off, where's my bound? Oh, I always get confused. I've got international. So you see, this is the if I change this to img, what it's gonna look, right? So this is a very like you can hardly see anything here, because as I was saying before, the images are really underexposed. And when we do this CMap thing, that's it does that, it extends the color and use the full range of dynamic range. So what we're doing here, loading up full image into a variable img. Now this img becomes an RMG, red, green, blue RGB image. So now we only take first channel of this image. So what does it mean? This, if you have this column, mean use all the rows, all the columns, and 0 mean first channel, and put it into this new object, new variable DNA. And now we show what this DNA look like. And this looks better because now we only extracted one channel. The values are still very underexposed. We're going to look that up in our histogram shortly, but the CMAP thing, it kind of increased the dynamic range. And so this is on our one channel, 0, 0 channel. On the first channel, on the second channel, we have our spindle. The spindle looks like this. Right? It's a ball-like structure having microtubules in there, which separates this DNA apart. And then on the third channel, we have our nucleus. If we put 2 here, right? So these are the nucleus and some noise in there. So this is a simple code to make histogram for our values. And if we run this, we can have a look like our values are really very much less than 10. Like we have a value of 1, almost more than like 50,000 pixels there, and really hardly any value greater than 20. Right, so that shows that the images are really underexposed, but you got what you got, and we have to work with them, and we can easily salvage our images using this thresholding methodology. And we can confirm that by this plot C color limits, by applying different color limits. We run it. For example, we define like V minimum, like our minimum color limit. I could put 2 here, or maybe let's put 10 here. And the max one, let's put 20 here. So you see, if we extract information greater than 20, we're going to lose a lot of information. Yeah, start from 20. And maybe go up to 100 or 110, doesn't matter. So there's nothing. Basically you see, now these will allow you to try different numbers and see where your region of interest is. What are the pixel values? So have a play with different numbers, 5 and 7. And I can now see that, yes, between these pixel value, that's where is my region of interest is. My region of interest consists of the values in between this range. So this is a manual way of just having a play with the different pixel values, just to see where is my region of interest. And then we can apply a global thresholding once we know some number. So it's a simple, you got your variable greater than 7, you multiply with a 1, put it into a new variable, and that becomes your mask. And you run it, you see this is our like a binary image. This is now our binary image. It has a clear defined boundaries, and this has the values of just a 0 and 1. So this is the global thresholding. I came up with this just a number, trial and error, using this method, looking at different range of numbers. Now, how we can come up with an automatic method? So we have got a few algorithms that can automatically give us a thresholding value. There are famous few methods already built into this library, skimage, and you can try all these big famous methods. So try all thresholding, run it, and we can have a look what our images look like. So this is our original image. If we do the ISO data, this is a algorithm, then our binary image going to look like this. If you apply Lee thresholding method, the image will be this, which is very bad. It has picked up a lot of noise. These are the noise in our data, because we know that from our knowledge of the domain, our region of interest just in the middle. If we apply minimum, then looking at back into this image, this is our DNA, and looks very much similar to this one, but still it's missing out if we closely look up this one. This UTSO, very famous thresholding method, it really worked well here. We can see it got rid of a lot of noise, and we have a very well defined region of interest. And the triangle and yen is not that triangle is probably is a bad yen is not that bad. Maybe it's very much similar to the UTSO. So UTSO, very famous one. Let's try this method. How do we do that? You just call this function thresholding. We visually inspected that the UTSO thresholding works well. Give it a DNA. It's gonna give you the global threshold number. You extract whatever is greater than that global threshold number, and that will make up your binary image. So we show that plot image. Now this is our binary image. Looks very good. Now we have extracted our region of interest now, but still there's a problem here. We have some noise in there. So now we're going to get rid of this noise. We can apply quite a few methods here to get rid of the noise. Erosion is one of the methods, but because I can see that my region of interest in this example is the biggest object in this image. So it's very easy to extract based on the size of an object. So my region of interest is the biggest in this image, right? So if I just say apply a morphology image, and then I'll say remove all the images which are below certain size. Let's say I just came up with this 25,000 number. It could be 20,000 number. It probably wouldn't matter, or 30 or 35,000 number. It will still be the same. So, and it's very easy. Now I got rid of all the noise, and now I left with only my region of interest. Right now, once I have this, my binary image, now it's very easy for us to calculate how many pixels are in there, what are the boundaries, what's the parameter, what's the center of this image. We can calculate all these properties from this binary image, and we can also go back to the original image and extract the actual pixel values. What do? You use this mask, put this mask on the top of the original image, and then you get rid of all the noise and the extra information that you have in the image. Yeah, so that's what we're doing here. Like this one is called DNA WB for black and white. So we make a copy of the original one, calling is DNA clean. So this DNA W1, yeah, our black and white mask image, put that into bracket, and this sign is negation. It's, and then we're saying equals to 0. That means that everything else which is not our region of interest will become 0. Yeah, so the DNA clean, which is a copy of the original DNA, it's gonna be, everything else will be 0. So that's why everything else becomes 0 except this region of interest. So this will allow us to calculate the intensities within this area. So black and white image, this binary image is good enough to do parameters, centricity, what's the shape, lots of important things that are related to this research. That's good enough. But to do any intensity-based experiment, to see if the intensity of a treated DNA is different from the untreated DNA intensity, so then we need to extract the original DNA. Other than that, for all the rest of the things, this one is good enough. Okay, now we have done what we're going to do. We're going to measure the parameter. And we use neighborhood 4. Remember, we discussed that in our lecture, that you have to define how your neighborhood going to be counted. Yeah, so your neighborhood could be 8, your neighborhood could be 4, and here we only using the 4. You'll get a slightly different answer. So we calculated the perimeter of this object. Similarly, we can calculate the total size of this object, which is about 46,000 pixels. And from the microscope, you also get how big is it. Once you have a pixel values, then you get a conversion formula from the microscope provider. So you can convert these pixels by multiplying, divide whatever the formula is, easily back into the nano centimeter, nano millimeter, or millimeter, or whatever. So we calculating here, we calculating like 4-5 properties of this object. Is we calculating the area, how big is that, parameter, what's the mean intensity, and what else? That's it. So we calculating these 4-5 things, and these are all available in using this SK image library again. So just printing these numbers here. Now this library has a lot of other things to calculate as well. All you need to do just to call this function. So one of the interesting thing I'll probably mention is eccentricity. Eccentricity tells you if your object is a circle or is an oval shape. So that's another interesting property that you might want to look at your object. And if your object was very circular before the treatment or after treatment, it become like an oval type shape. So that's the number gonna give you that. So, and then the mean intensity, lots of other type parameter type things, solidity, how the object is, how solid is that object. So lots of properties you can calculate using these functions. And then you can compare all of these property of treated against untreated images. Right. Now we have defined our pipeline. We first you do all this testing on a single image. You do test your code, and then you run it through, pump through all the files, and then do that calculation. So here we defining that, processing that images, taking the folder, and it basically copy paste all the above code within this function, and it going through the folder and processing all the images and saving into a CSV file. So we could have two different folders for CSV and untreated, or you could have a one CSV file for both. Then you need a label at the end. So in this example, we're creating two files. One is calling untreated. We process untreated folder, and then you run the same code again, pass it the untreated, pass it the treated folder, CIC, which is CIC knocked off. And it's going to create you a second CSV file. Now you have two CSV file. Now to apply deep learning or machine learning algorithms, what you need to do, put these two files into one, and then create a new column there called label, and put that manually label there for all the file, all the information that is coming from untreated, you label it as untreated or 0, and all the information that coming from treated file, you label it as a 1 or call it treated. Right, and then now you have a big CSV file. You do the machine learning, deep learning, whatever you do, and you can also do statistical analysis, and that will allow you to quantify objects and also give a meaningful information that this, you can show the efficacy of a drug or a treatment with some numbers, with some statistical analysis. You can still apply deep learning approaches to do the classification that yes, this is the untreated, this is a treated for, but you see, it's not really meaningful. You can, if you say, I've got a deep learning algorithm which gonna tell you whether this image is treated or untreated. Okay, what does it mean? Does it mean anything? It doesn't mean anything. Yeah, unless you give this information to your authorities, to your researchers, that this is untreated and this is treated. Why? Because the size has gone bigger or size has gone smaller, parameters has been changed, its shape has been compromised, it has been decomposed, or whatever, then it's a meaningful information. And that you also apply that statistical analysis on top of it to give it more like a meaningful reasoning. And then the authority is gonna kind of will believe you that what you're claiming could be right. Okay, does it make sense? Any question here?